Every morning you perform a routine. You wake up at a certain time. You do certain things in a certain order. You eat certain foods, or you skip eating altogether. You move, or you do not move. You expose yourself to light, or you stay in the dark. You manage your stress, or you carry it into the day. None of these feel like medical decisions. They feel like habits, the unremarkable background of a normal morning. What the cancer biology research of the last 15 years has established, with a specificity and consistency that has surprised even the researchers conducting it, is that this unremarkable background is not neutral. The molecular environment your morning routine creates in your body is either suppressing tumor cell activity, activating immune surveillance, clearing pre-malignant cells through autophagy, and maintaining the epigenetic programs that prevent oncogene expression, or it is doing the opposite. Every morning, without exception. And the difference between a morning routine that is fighting cancer at the cellular level and one that is feeding it comes down to 5 specific biological switches that your habits are toggling, knowingly or not, before most people have finished their first cup of coffee. I want to be precise about what I mean by fighting and feeding, because the framing matters. I am not describing morning routines that cause cancer or cure it. I am describing the molecular biology of cancer suppression and cancer promotion, the cellular environment that makes it easier or harder for pre-malignant cells to survive, evade immune detection, acquire mutations, and progress toward clinical malignancy. Every adult over 50 has pre-malignant cells in their body. That is not alarming. It is a documented biological reality. Your immune system identifies and destroys most of them before they become anything. The question is whether your morning routine is supporting that immune surveillance and cellular housekeeping, or suppressing it. That is the distinction that the research is increasingly able to quantify. I am Dr. Edward. What you are about to hear is educational information based on published scientific research. It is not a substitute for personalized medical care. Nothing in this video is intended to suggest that morning routines replace cancer screening, treatment, or medical supervision. I am describing modifiable lifestyle factors that have documented effects on cancer biology, factors that exist alongside, not instead of, the medical care that every senior should be receiving. Subscribe to this channel right now. Before we go further, this video connects research from oncology, immunology, circadian biology, and metabolic science in a way that I have not seen presented together before, and the connections change how you understand what is at stake in your morning. Subscribe now, it is free, and stay with me. Let me start with the biological framework that makes a morning routine oncologically relevant, because without this framework, the individual habits I am going to describe look like general wellness advice rather than the specific cancer biology interventions they actually are. Cancer is not a single event. It is a process, a multi-step progression from a normal cell through a series of genetic and epigenetic changes toward a cell that divides uncontrollably, evades immune destruction, and eventually forms a clinically detectable tumor. This process takes years to decades in most cancers. It requires the accumulation of multiple enabling conditions: DNA damage that evades repair, epigenetic silencing of tumor suppressor genes, activation of growth-promoting oncogenes, suppression of the immune cells that would otherwise destroy the abnormal cell, and a local tissue environment that supports rather than limits tumor cell survival. Each of these steps is influenced by the molecular environment the cell lives in, an environment that your morning routine is shaping through 5 specific mechanisms. The first mechanism is cortisol and immune surveillance timing. Your immune system does not run at constant capacity throughout the day. It follows a circadian rhythm, a 24-hour biological clock, that is orchestrated primarily by the circadian timing system in the hypothalamus and entrained by light exposure in the morning. Natural killer cells, cytotoxic T lymphocytes, and the dendritic cells that coordinate immune responses all peak in their activity and circulation during specific windows of the day that are aligned with the morning light signal. When your morning light exposure is appropriate, when you receive natural or bright artificial light within the first 30 to 60 minutes of waking, your circadian clock is precisely calibrated, your immune cell cycling is synchronized, and the morning peak in natural killer cell activity that represents your body's primary defense against pre-malignant cells occurs at the right time and at the right magnitude. Research published in the Journal of Experimental Medicine found that natural killer cell activity follows a circadian pattern with a peak in the early morning hours, and that disruption of this circadian pattern, through light exposure at the wrong times, through irregular sleep-wake timing, or through morning routines that delay light exposure to the eyes, significantly reduces peak NK cell activity. NK cells are the immune system's frontline cancer surveillance mechanism. They identify cells with abnormal surface markers, cells that have lost the MHC class I expression that healthy cells display, and destroy them before they can proliferate. A morning routine that begins with curtains drawn, phone screens in darkness, and no natural light exposure until mid-morning is a morning routine that is running its NK cell peak at diminished capacity every day, compounding over months and years. The cortisol component compounds this. Cortisol follows its own circadian pattern. It should peak sharply within 30 to 60 minutes of waking in what is called the cortisol awakening response, then decline throughout the day. This peak is not purely a stress response. At its normal physiological magnitude, the morning cortisol peak has an important immunomodulatory function. It calibrates the inflammatory response and optimizes the balance between pro-inflammatory and anti-inflammatory immune activity. When the morning cortisol peak is blunted, as it is by chronic psychological stress, by poor sleep, by irregular waking times, and by the HPA axis dysregulation I described in my hormones video, the immune system's anti-tumor activity is compromised. The NK cell peak loses its calibrated timing. Inflammatory cytokines that normally suppress tumor microenvironment formation are not adequately produced, and the tumor-promoting inflammatory environment that chronic unresolved stress creates begins to dominate. The second mechanism is insulin signaling and tumor cell fuel availability. I described in previous videos how chronically elevated insulin from continuous eating patterns, from high-glycemic diets, from insulin resistance, suppresses AMPK, activates mTOR, and prevents autophagy. These same pathways are directly relevant to cancer biology through a mechanism called the Warburg effect. Cancer cells, unlike normal cells, preferentially metabolize glucose through glycolysis even in the presence of adequate oxygen, a metabolically inefficient but extraordinarily rapid fuel strategy that allows rapid cell division. Cancer cells are, in metabolic terms, glucose addicts. They upregulate glucose transporters, compete aggressively for available glucose, and respond to elevated insulin and IGF-1 signaling, the hormonal consequence of chronically high blood glucose and insulin resistance, by activating PI3K-ACT-mTOR pathways that directly promote cell survival, proliferation, and resistance to apoptosis. A morning routine that begins with a high glycemic breakfast, the traditional combination of juice, cereal, toast, and fruit that most seniors were taught is healthy, spikes blood glucose, elevates insulin, activates the PI3K-ACT pathway in every cell, including any pre-malignant cells present, and provides the glucose surplus that Warburg-metabolizing cancer cells preferentially consume. A morning routine that maintains the overnight fast through the early morning hours, as I described in the longevity switch video, keeps insulin low, activates AMPK, initiates autophagy that clears pre-malignant cells, and deprives any tumor cells present of the insulin and glucose surge they are most adapted to exploit. A study published in Cancer Research found that overnight fasting of greater than 13 hours was associated with a significant reduction in breast cancer recurrence risk in early-stage breast cancer patients, with a proposed mechanism involving reduced IGF-1 signaling and increased tumor cell autophagy during the extended fasted state. Subscribe to this channel right now. I have 3 more mechanisms to give you, and they connect everything I have been building in this series into a single unified picture of what your morning is actually doing. Subscribe. It is free, and let us continue. The third mechanism is reactive oxygen species balance and DNA repair window. Your cells accumulate oxidative DNA damage throughout the day, from metabolic processes, from environmental exposures, from the byproducts of inflammation. The repair of this damage occurs primarily during sleep and in the early morning hours, driven by growth hormone pulses and the circadian expression of DNA repair enzymes that peak during the sleep-wake transition. The quality of this repair window, how completely and accurately the accumulated DNA damage is corrected before the cell divides again, determines how many mutations persist in the genome of that cell and its daughters. A morning routine that chronically disrupts the repair window, through inadequate sleep that truncates the overnight repair period, through morning alcohol consumption that generates reactive oxygen species and competes with repair enzyme activity, through heavy exercise in the immediate waking period before repair is complete, or through the morning cortisol dysregulation I described, leaves a residue of unrepaired DNA damage in every cell that has divided that day. Over months and years, this residue accumulates. The cells that have sustained the most unrepaired damage are the ones most likely to acquire the oncogenic mutations that characterize cancer initiation. A morning that protects the repair window, adequate sleep duration, gentle rather than strenuous early activity, no morning alcohol, and the light exposure that calibrates the circadian repair gene expression, is a morning that starts the day with a lower mutation burden than the morning that disrupts it. The fourth mechanism is the gut microbiome and immune priming in the morning. The trillions of bacteria that inhabit your intestinal tract are not static passengers. They are active immunological educators. They produce short-chain fatty acids, communicate with intestinal immune cells, and influence the systemic immune response in ways that are increasingly recognized as relevant to cancer biology. The gut microbiome has a circadian rhythm. Its composition and metabolic activity fluctuate over 24 hours in patterns synchronized with the host's feeding, sleeping, and activity cycles. The morning feeding window, the first meal of the day, is one of the most potent modulators of the daily microbiome cycle. What you eat first in the morning and when you eat it shapes the bacterial populations that dominate the subsequent hours. Research published in Cell has found that the gut microbiome composition of cancer patients differs significantly from healthy controls in ways that affect both tumor progression and treatment response. Specifically, the abundance of certain short-chain fatty acid-producing bacteria, particularly Akkermansia mucinifila and Faecalibacterium prausnitzii, is inversely associated with colorectal cancer risk and positively associated with immunotherapy response. These bacteria thrive on fermentable fiber. They are suppressed by high-fat, low-fiber, high-sugar morning meals. A morning routine that begins with soluble fiber, oats, legumes, ground flaxseed, fruit with the fiber intact, provides the substrate that immunologically beneficial bacteria require to maintain their populations through the day. A morning routine that begins with highly processed food or no fiber at all begins a day of microbiome composition, drift toward the bacterial populations associated with increased gut permeability, systemic inflammation, and the tumor-promoting inflammatory environment that oncologists associate with cancer progression. The fifth mechanism is methylation and epigenetic gene expression in the morning hours. This is the most complex mechanism I am going to describe, and also the one with perhaps the most direct connection to cancer initiation. DNA methylation is one of the primary epigenetic mechanisms by which gene expression is controlled. Methyl groups attached to cytosine residues in DNA promoter regions silence the genes they mark. Tumor suppressor genes, the genes that limit cell proliferation, promote apoptosis, and maintain genomic stability, are silenced in most cancers through aberrant methylation of their promoter regions. The cancer does not need to mutate the tumor suppressor gene and destroy its function. It simply needs to methylate the promoter and prevent the gene from being expressed at all. The methylation patterns in your cells are not fixed. They change in response to dietary methyl donors, to inflammatory signals, to metabolic conditions, and to the circadian timing of the enzymes that add and remove methyl groups. A morning that provides adequate methyl donors, folate from leafy greens, B12 from eggs or animal products, betaine from beets and spinach, choline from eggs, supports the methylation capacity that maintains normal tumor suppressor gene expression. A morning that begins in chronic inflammatory conditions, from poor sleep, from high cortisol, from a high-glycemic breakfast, promotes the inflammatory cytokine signals that aberrantly activate DNA methyltransferases and silence tumor suppressor promoters. Research published in Cancer Epidemiology, Biomarkers and Prevention found that dietary patterns with adequate methyl donors were associated with measurably reduced methylation of the p16 tumor suppressor gene promoter, a methylation event that is one of the most common initiating steps in multiple cancer types. I also want to address something specific about sleep timing in cancer that most people find surprising. It is not just sleep duration that matters, it is sleep timing, specifically whether your sleep occurs in alignment with darkness or in opposition to it. Research from the National Cancer Institute examining shift workers, people whose sleep occurs during daylight and whose waking hours occur at night, found that long-term night shift work was associated with significantly elevated risk of breast cancer, colorectal cancer, and prostate cancer, classified by the International Agency for Research on Cancer as a probable human carcinogen. The mechanism involves suppression of melatonin, which I described in the cellular aging video as a direct activator of the SIRT1 longevity sirtuin and a powerful inhibitor of mTOR. Melatonin also has direct anti-tumor properties. It inhibits aromatase, the enzyme that converts androgens to estrogens in adipose tissue, and that is a target of the aromatase inhibitor drugs used in breast cancer treatment. It suppresses the WNT beta-catenin signaling pathway that drives colorectal cancer proliferation, and it directly inhibits the tumor neovascularization that allows tumors to develop their own blood supply. A morning routine that ends sleep in genuine darkness, that protects the melatonin synthesis of the final sleep hours by avoiding light exposure until the appropriate morning wake time, is providing these direct anti-tumor effects every single night. Now, before the action framework, this is the moment to share this video, because the person who needs it is not the person already thinking about cancer prevention. It is the person whose morning routine has not changed in 20 years, who believes their habits are either healthy or irrelevant, and who does not know that the molecular biology of cancer promotion and suppression is being shaped by the hour before they leave the house. Share this with them now while the 5 mechanisms are clear in your mind. Then stay for the action plan, because the specific sequence of the morning and what goes in it is what converts this biology from information into protection. Here is what to do in the next 48 hours based on what you just learned. Tonight, set a consistent wake time and commit to it for the next 2 weeks. Circadian immune surveillance depends on regularity more than on any other single variable. An irregular wake time, varying by more than 30 minutes from day to day, is enough to desynchronize the NK cell peak and blunt the cortisol awakening response that calibrates morning immune function. Pick a time, hold it. Tomorrow morning, within 30 minutes of waking, expose your eyes to bright natural light for 5 to 10 minutes, not through glass, through an open window, or by stepping outside. If your wake time is before sunrise, use a 10,000 lux bright light therapy box for 10 minutes. This is the signal that calibrates everything else. The circadian clock that times your immune peak, your cortisol awakening response, your DNA repair gene expression. It costs nothing and takes 10 minutes. Tomorrow breakfast. Extend the overnight fast to at least 12 hours from your last meal before eating. When you do eat, begin with something that does not spike blood glucose and does provide fermentable fiber, a handful of mixed nuts and a piece of whole fruit, or eggs with leafy greens and a tablespoon of ground flaxseed. These are not arbitrary healthy choices. They are specific inputs that keep insulin low, feed the short-chain fatty acid-producing bacteria, and provide the methyl donors that maintain tumor suppressor gene expression. This week, audit your morning alcohol. Any morning alcohol, wine at brunch, a Bloody Mary, a mimosa, generates reactive oxygen species during the circadian window when DNA repair enzymes are completing the overnight repair cycle. This is not a restriction I apply to all alcohol at all times. It is a specific concern about the morning repair window. Morning alcohol is the timing most directly disruptive to the DNA repair process. You now know that your morning routine is not neutral. It is a set of 5 biological switches that your habits are toggling every day. Immune surveillance timing, insulin and tumor fuel signaling, DNA repair window integrity, microbiome immune priming, and epigenetic tumor suppressor gene expression. Each switch has a position that suppresses cancer biology and a position that enables it. The position of each switch tomorrow morning is a choice you are now equipped to make. Subscribe to this channel if you have not already. My next video covers the specific foods with the strongest evidence for direct anti-tumor effects, the compounds, the mechanisms, and the doses that clinical trials have found meaningful. This is the dietary companion to everything I told you today. Subscribe so it comes directly to you. I am Dr. Edward. Your morning is a medical decision. Start making it one. I will see you in the next one.